Hi, everyone. I'm Jack Cush with Room Now. I am joined by the experts. This is a roundtable on spondyloarthritis. I'm going to ask everybody to introduce themselves. I'm Jack Cush in Dallas, Texas. Alexis.
Hi, I'm Alexis Ogdie. I'm Associate Professor of Medicine and Epidemiology at the University of Pennsylvania in Philadelphia.
Chris.
Christopher Ritchlin, Professor of Medicine at the University of Rochester in Rochester, New York.
And Dr. Scher.
Jose Scher, New York University, New York City, rheumatologist.
All right, so the meeting is over and done with. These folks are still standing. It's amazing. If you only shadowed them this week, you'd be astounded by the amount of work that they put in. So I'm most thankful that you're here and wanting to share your experiences or what you thought were good from the meeting as far as content. Let's start with Alexis. Give us something that you thought was worth remembering.
Well, I think one of the new things that we saw this meeting compared to other meetings was the introduction of Brepiscipnib. So this was a phase 2 study comparing Brepiscipnib, which is a TYK2 JAK1 inhibitor, compared to placebo in a phase 2 study, so dose ranging. And they were examining whether or not this was a worthwhile therapy in PSA. And it looks like it was doing okay from an ACR 20 perspective. From a safety perspective, actually looks similar to JAK1, but no significant dose effect. That was the one difference from our other JAK1s that we've seen.
So do you think that this is different than a JAK inhibitor? You know, current JAK inhibitors knock out more than one JAK. They like to say that they're doing one, and hitting TYK and JAK together, does this make it really unique?
Well, it could be unique. So I think one of the things that we're trying to figure out is where would this be placed? So we have a TYK2 coming for psoriasis and psoriatic arthritis, and it appears that it works pretty well. Safety looks pretty good. We have a JAK1 coming for psoriatic arthritis, already available for RA, also coming for atopic dermatitis. So, you know, I think we still have those other two on the horizon yet. So this is a kind of behind them. So I guess it'll be interesting to see where that falls into place once it's finally available.
All right. Anybody else have any feelings about TYK2 as a target in Spondy or PSA? Is it going to be in there?
It makes sense from the point of view of the model, and the model is that TYK2 is involved in the IL-23 pathway. And one would say that seems to be confirmed clinically, at least in psoriasis, where the oral Brepiscipnib is pretty effective for psoriasis. I'm not overwhelmed by its efficacy in psoriatic arthritis, though, nor am I overwhelmed with the data that Alexis just mentioned. So I'm not sure about the arthritis at this point.
Okay.
And you were there, Jack, on the FDA recap of the year. You know, I think that's the elephant in the room. Are they going to consider it a JAK? And if so, do they get the label? And if they do they have a place for any rheumatic condition?
Yeah.
Remains to be seen.
The senior director saying, where's that going to fall? And he said, well, it's in the family. And he didn't, of course, he didn't actually truly answer the question, but clearly he's going to be considering, the FDA is going to be considering that under the same umbrella as all the JAKs when it comes to safety. And that's another hurdle for them to overcome in drug development.
JAK, recently a topical JAK for psoriasis was released. It has it has black boxes for the topical. So I think that also says a lot, right?
Yeah. Interesting that they would give the box warnings to the topical, but not put that on ruxolitinib and the other JAKs that are out there for maybe other indications. Yet the mechanisms are still the same. FDA's rationale on that, of course, was that these other Bari, UPA, TOFA all have the same indications, chronic inflammation, same target audience. That's why they get that slap with that. I don't know. It seems arbitrary to me. Chris, why don't you give us your next one?
Yeah, so two of the major challenges in axial spondyloarthritis are delay in diagnosis and trying to get a handle on how to read the plain radiographs and MRIs for the presence of, in the case of X-rays, sclerosis, narrowing, etc. And in the case of MRIs, what the heck is bone marrow edema and how can we use that to diagnose this disease? And there were a number of abstracts dealing with the radiographic and MRI that were somewhat interesting, and they were using artificial intelligence and machine learning to have a group of experts get together, 3 in one case, to interpret these films. And then from the variables that they used, they built these models using artificial intelligence and machine learning. And they showed that these were pretty effective at recreating what these expert readers have done. And they did this. These were different abstracts. I'm combining them. One was with plain radiographs, and that was done by Dennis Badobny and his group. And then there were a couple that looked at MRI using these strategies. I think this is encouraging because we know that in many hospitals, the radiologists are not used to reading these kinds of films, and they're not particularly good at it. And so I'm thinking we all talked among our group that maybe there'll be like a centralized place that these get sent to, and you get a reading score and it comes back to you, and it may be fairly accurate and valid. The second is, and I'm going to need Alexis to help me with this, and that was trying to decrease the time to diagnosis of AxSpA. And in the UK, they have a large database, which they used a technique to try and develop algorithms that would allow more effective referral to rheumatologists for patients with axial SpA. Alexis, you want to just tell them quickly? I'm not good at this one.
Yeah, so they took in CPRD, which is basically an epic across the UK, taking a bunch of practices from the UK. They looked at patients with AxSpA and patients who were matched on other characteristics. And then they looked at what was happening in the years right before AxSpA to see if there were some codes that kept coming up that would potentially predict the patient who's likely to develop AxSpA. So then they use machine learning to train these algorithms to identify differences between the two groups. And the idea is that they've had this algorithm that works well now. Now they want to kind of put this into a prospective manner and say, even if it's just a cohort study, if you start with all patients, can this predict who's going to go on to develop AxSpA, or who may even just need to see a rheumatologist because they may have AxSpA or might have similar symptoms. So it's something that's been going on in terms of PSA as well. I think these things have been mainly in this case control design where you're selecting people with and without the diagnosis and looking back in time. But now we really need to take this forward in time to see how well do they actually work in the real world.
And do we have results, or we're still at the stage of now testing it?
Developing. Yeah, we're still at the stage of developing algorithms in a case control type design.
It's brilliant, because most rheumatologists are surprised when they find out that we're not the ones diagnosing ankylosing spondylitis and spondyloarthritis, that they're being diagnosed incidentally or wrongly, or God knows, a million different ways. And we get involved, which means we get the patient way too late. Actually, we presented an abstract this time looking at that. And we were, so surprised to find that there's so many of our AxSpA patients are in primary care. There's like 10 times as many patients that with AxSpA in primary care compared to rheumatology. There have been some reports in some centers where they have developed a scheme for earlier referral, and they've shown pretty good numbers, getting away from that 7 to 9 years of that to make a diagnosis, and done it much better. But this sort of takes it out of the hands of intelligence and goodwill, and start, using AI to earmark people who should be referred, should be tested, that sort of thing. Really brilliant. I like that, Chris. Thanks. Jose, do you have one?
I have one. Actually, I was listening to Alexis and Chris. We try and diagnose earlier and diagnose better. And yet what strikes me today, and part of it is data from Alexis's group, is up to 40-some odd % of our patients with spondyloarthritis are on opiates. That's striking to me. How are we connecting those dots? We're going to try and make the diagnosis earlier. We're going to try and make them better. We have significant improvements in terms of mechanism of action, new medications. We're talking TYK2 now. We're all happy and excited. And then we see data on 40% of people on opiates. It is astonishing to me that we're there, even after the scandals that are emerging or have emerged. Who's prescribing those? Why are we prescribing those? And what do we do about it?
So isn't that two issues? One, pain, and pain was a big subject at this meeting, right? In SPA especially. And two, as you say, Jose, who's making the diagnosis? Alexis, you had an abstract on that. You want to share?
Yeah, we examined these patients in primary care as well within our health systems. We've actually done with 3 health systems. We're going to put it together to look across different health systems. But in this particular abstract, we just looked within Penn, and we said, let's take all patients with ACSPA who've been seen between 2017 and 2020. So this is not even like 10 years ago. This is like now. And look at who's seeing them on a regular basis, and who's prescribing therapies for them, and what therapies are they prescribing. So we looked at CSD MARDs, biologic therapies, opiates, and non-opiate pain medications. And there's huge discrepancies in the types of therapies that patients are getting depending on who is prescribing the therapies. So primary care providers are prescribing a lot of opiates still for our ACSPA patients, but they're also prescribing more non-opiate pain medications, suggesting they're kind of dealing with the brunt of the pain aspects. And then we obviously prescribe a lot more biologic therapies than the primary care doctors would. But like Jose said, it really speaks to how far we have to go, because even though we're getting diagnosed with these patients, they're clearly not getting to us and getting the appropriate therapies. But also that reverse piece, which is how do you deal with the pain? Opiates, we know, are the wrong answer for the way to deal with pain. They're also easy. In some sense, it's getting harder all the time. But maybe we need to rethink how we think about this disease and separating the pain component too, and how do we manage that pain component?
I guess the other question, though, Alexis, is what percentage of those patients that were being treated with opiates were not getting biologics that should have been getting those, right? I mean, if you got a patient who's on max biologics and they're having horrible pain, that's different than a patient who's getting opiates and has ACSPA and is not getting treated for the inflammation. And that's probably more common, obviously, in the primary care world, right? We hope.
Or NSAIDs for that matter.
Or NSAIDs, right.
Right. I mean, we don't need to go to the biologic. NSAIDs is a perfectly good treatment for ACSPA.
Right.
Right.
Like going to the dentist, getting a tooth pulled out and getting, 7,000 grams of opiate. So no need.
Yeah.
Advil is just good.
Exactly. That's a good question.
Ibuprofen, excuse me, Dr. Cush.
Are there any new insights on pain, though, coming from the meeting?
There's a lot of pain and it affects your outcomes. But I think that's as far as we've gotten. I don't think we've done a good job of addressing how to deal with the pain. There was that cannabinoid study, but that didn't really provide an answer for how do you, didn't seem to do much, at least.
It was as good as placebo.
In years past, and even this year, we see a lot on comorbidities, but I saw more this year. Maybe it was just the Twitter feed on work outcomes. Was that novel or is that informative?
It's not, no.
Yeah. Okay. Good enough. Jose, you did this study about microbiome studies in monozygotic twins.
Oh, yeah.
Tell the audience about that.
Yeah, sure. So I think in some of I think Christopher's contributed a pair. It's very hard to find, as you may know. Discordant twins for psoriatic disease.
Wait, let me say discordant meaning the
one affected, one unaffected.
Right.
Right. Okay. And so it takes time. We have up to 16 right now. So what we're doing a variety of things. A unique cohort, 16 pairs, right? So we started off with the microbiome, the ACR1. We're seeing a lot of differences right now, particularly in the skin, in the non-lesional skin of people with psoriatic disease. They have a lack of diversity that's very similar to the plaque psoriasis. Does that make sense? A non-lesional skin on someone with psoriatic disease, it's similar, microbially speaking, than that of someone, or the same plaque on the same patient. And then if you look at the normal skin, it's completely, robust and healthy. Same or something similar happens in the gut microbiome. There's that decrease of diversity, what we call dysbiosis, and some of the metagenomics, some of the genes that these microbes intrinsically have are helpful for maintenance of health, are lower or even absent in the affected twins. So there's a lot to discuss there. There's been a lot of work on how you reconstitute that. Is it like a microbiome approach? Is it a probiotic approach? Is it a dietary approach? Alexis has a grant and a study ongoing to compare two different dietary approaches in psoriatic disease. So it's in the making, and hopefully this is informative.
Jose, since you're, bow deep in this problem, do you think at this point you could say that these microbiome unique profiles that we've found, you think they're more important in disease pathogenesis, disease activity, or drug responsiveness, as you've shown?
Yeah, we're doing that now. Yeah, I think it makes a lot. I mean, certainly it's true for pathogenesis when you do animal models, right? That's established and proven over and over again. Does that remain true in humans? We have not seen that much direct evidence that that's the case. When it comes to its relationship with the drugs that we prescribe, methotrexate, sulfasalazine has been the case forever, right? But now we're seeing evidence in not only methotrexate, which we've been doing work on, but also biologic therapies. This is data mostly from the cancer literature and from the IBD literature showing that certain baseline microbiome predicts the response to future therapy. And so I think that's where we're heading. The other one, we need a lot of mechanism in the humans, because one thing is to sack mice and just say, these microbes elevate Th17 cells or whatever, TNF. But to prove it in humans will be difficult. AMP and other endeavors that are coming up will be helpful to try and understand and get us some answers there.
Well, since I have three experts here in spondylitis, can I ask a question? I'm a little confused about IL-23 and spondylitis. Supposedly it doesn't work, but yet there's this guselkumab showing that it works in psoriatic spondylitis. And then you got the JAK inhibitors showing that they work in spondylitis patients, ankylosing spondylitis. So is 23 out of the picture, or is this, what am I to learn from all this?
So, Jack, I think we have to be careful about over-interpreting the results that we're seeing in peripheral arthritis trials in regards to axial outcomes. So we saw the Discover 1 and 2 sort of post hoc analyses, and we heard at this meeting from Xenophon Baraliakos, who did a post hoc analysis of the in psoriatic arthritis of the select trials that were used to treat peripheral PSA with UPA, using a similar approach, although they didn't have any imaging data, they just looked at PROs. And we've had a lot of discussions about it, the three of us, and our group with Eric and Laura as well. And it's really hard to say that BASDI or even ASDAS is telling you something specifically about the axial skeleton in a very strongly peripheral arthritis group. I think from a biologic point of view, there's pretty strong evidence that IL-23 may not be involved in ongoing axial inflammation, but may be instrumental in starting the whole process, getting it going underway, and that there are many different cells that are present in the spine and the entheses that can release IL-17 independent of IL-23, and mainly of the innate type. So I think the jury's out, to tell you the truth. You have to do the imaging study like they did Maximize with secukinumab, which I know they're going to do. I think they are. And then you'll really have a sense of whether or not blocking IL-23 really has axial legs in psoriatic arthritis axial disease. And you guys I'm sure you have other thoughts, but.
I think that nicely summarizes my thoughts too. I mean, I think the critical thing is to not rely on the BASDI as a measure of axial disease in PSA, because it's so conflated with the peripheral disease. So I think those studies are nice, but they're hypothesis generating and need to be followed up with imaging.
So can we end on some optimism other than Brepiscipnib maybe coming on board? Are there other reports that we can be optimistic about? Biomarkers, profiling, predictive variables. Anybody have any suggestions from the meeting?
We're working hard. I don't know, it's part of the meeting, but it's been suggested throughout the meeting, and that is the question of why we're doing monotherapy. Why is it that we're still in that era of either one cytokine or pathway or the other? Where is the space for, one molecular pathway that addresses the spine, let's say, and one other molecular pathway that addresses the skin domain? That may be the future. There are studies in the making, not part of this meeting, but probably the next one.
A clinical interjection here that's maybe helpful to the audience. When I've had to deal with, prior authorizations, and I got patients on 2 biologics, which I have done, the greatest thing is to say, well, one drug, ustekinumab, is for this, and the other drug, the TNF inhibitor is for that, because I'm treating 2 different diseases. And sometimes that, in fact, works. Chris, you were going to say.
No, I totally agree with what Jose said. There was one abstract that did not receive a lot of attention. And, there's been a lot of discussion about whether or not axial SpA is a disease of innate immunity or of the acquired immune system. Is it like RA where there's an autoantigen? And so far, that's not been something we've been able to show. But there was an abstract from Matt Brown's group in Australia where they took a look at patients that are HLA-B27, and they were able to isolate CD8 cells, which are tied to axial SpA, that what we call clonally restricted, meaning that they express T cell receptors that are specific for one antigen. And they had many of these that were of the same clone from patients that only were HLA-B27 positive. And so these data are really exciting, and using new technologies, you may be able to identify these clonotypes that are reacting to a specific antigen. And indeed, it's possible that axial SpA is an antigen-driven disease. And they also found this in patients with reactive arthritis. So we're back to the microbiome again as a driver of possibly an autoimmune response. And I think that's really exciting, and I'm really following that closely.
Do you think such patients with a clonally restricted response are more likely to have better targeted therapy?
I think that there might be ways to address the antigen driving component of it, right? So if it is microbiome, if it is infection, et cetera, you might be able to intervene earlier. Obviously, that's pie in the sky, but it's something that is very important. And I know you're covering RA, and there's really cool work in that, and in terms of the microbiome as well. So I think we're getting closer and closer to the source. Let's put it that way.
Great. Alexis, you have one more you want to end on, or are you happy?
I'm happy. I guess I'll just slightly add to Chris's comments, in which Chrissy Kuhn also had some nice studies looking at trafficking of cells, and that would kind of add to the story that Chris just kind of discussed. So some other kind of things that have been developing over the last several meetings, actually. So kind of interesting to see where that might.
Excellent. All right, folks, you're still standing. I'm amazed. I'm most thankful for your insights and teachings. We'll see you at the next ACR face-to-face in Philadelphia.
Thanks, Jack. Thanks from now.
Thank you, Jack.
Good one.
Thank you.
Nice to see you.